Diabetes and Insulin Resistance by Phoebe, Naudia, and Katya Williams. Think about the last group project you've done. You and the other members of your group had to talk to each other in order to complete the project, right? Well, just like the members of a group need to communicate to get the job done, the cells in your body talk to each other in order to keep you happy and healthy. This communication is called cell signaling, and the words cells use are really specific chemicals called ligands to tell each other what to do. Cells hear the chemical words through the signal transduction pathways. First, the target cell detects the ligand signal outside the cell. The ligand binds to a receptor on the outer surface of the cell membrane in reception. The receptor initiates the second stage of signal transduction, a signal transduction relay. One such relay system is a protein kinase cascade, named for the protein kinase, an enzyme that modifies other proteins by adding a phosphate group. This is called phosphorylation, and it only works because of cyclic AMP, which is constantly converted to ATP for protein kinases to steal phosphate from. Each protein kinase takes phosphate from ATP, turning it back into cyclic AMP, and phosphorylates another protein kinase, causing it to repeat the process until kinase modifies a signaling molecule inside the cell. This is the third and final step of signal transduction, the cellular response. An intercellular signaling molecule from signal transduction pathway produced causes the cell to perform a specific action. In the case of a hormone insulin, the insulin bonds to a G protein linked receptor, or GPCR, which bonds with a G protein to link it to an energy-rich GTP molecule. An activated G protein then activates the first protein kinase, which eventually leads the cell to more GLUT4 transporters in the cell membrane. Now we know how cells receive signals, but we don't know from where. There are actually two different wheres: local and long distance. In local signaling, a cell secretes a signaling ligand into extracellular fluid from which nearby cells can receive the chemical. If cells could talk, it would be like having a face-to-face conversation. In long-distance signaling, however, the cells are too far apart to directly talk to each other, so they use the bloodstream as a molecular telephone. A gland releases a hormone into the body's blood vessels, where it cycles around the body until it reaches a cell with the special receptor necessary in order to answer the chemical phone call. But what does any of this have to do with diabetes? Well, diabetes mellitus is a disease caused by broken cell signaling loops, making it difficult or impossible for cells to take in glucose. Normally, when blood sugar levels rise, the pancreas detects a change in homeostasis and secretes insulin into the bloodstream, where it bonds to cells with certain GPCRs. The GPCR then sets off the domino effect that eventually leads the cells to take up more glucose until blood sugar levels rise and return to normal. In people with diabetes, however, the pancreas either produces no insulin or the cells do not respond to insulin. There are two main types of diabetes, type 1 and type 2. Type 1 diabetes occurs when beta cells in the pancreas stop producing insulin. Research hasn't pinpointed the direct causes of type 1, but the risk of developing this type of diabetes is increased by certain variants of the HLDQA1, HLDQB1, and HLA-DRB1 genes, which belong to the human leukote antigen complex in chromosome 6. Leukocytes, or white blood cells, produce antibody proteins to kill foreign invaders such as viruses and bacteria when leaving other body cells unharmed. In people with type 1 diabetes, though, the mutated leukocytes produce autoantibodies, attack proteins turned traitor, that damage insulin-producing beta cells in the pancreas. In these cells, impairs insulin production and leads to type 1 diabetes. Although usually diagnosed in children and young adults, these malfunctioning genes are only expressed if a trigger prevents itself, like cold weather, a virus, or early diet. The mutated alleles are also recessive and most common in people of white ethnicity.
Type 2 diabetes is more common than type 1, although easier to prevent due to a stronger correlation with family history and unhealthy habits. 12 main genes have been linked to type 2 diabetes. The insulin gene INS encodes the shape of the hormone. If even one amino acid and the sequences change, the protein will not perform its job. Receptor coding genes, such as ABCC8, GCGR, and INSR, affect the ability of a receptor protein to bond to its ligand and initiate signal transduction. Enzyme coding, transcription factor, and regulatory subunit genes like CAPN10, GCK, LPL, HNF4A, PPARG, and PIK3R1 affect the function and efficiency of the signal transduction relay. Finally, we have the glucose transporter gene, GLUT2, which dictates the shape and therefore effectiveness of a glucose transmembrane protein. Type 2 genes are located across multiple chromosomes and are linked to both dominant and recessive traits. Mutated expressions of these genes contribute to insulin resistance, or the inability of cells to respond to insulin. Insulin resistance causes glucose to build up in the blood and interfere with bodily function. Those afflicted with diabetes usually experience dehydration, blurry vision, weight loss, tingling and numbness in the hands and feet, and irregular urination. People with both types of diabetes will exhibit dangerously high blood sugar levels, which causes the nerve damage responsible for tingling, numbness, and vision loss. But people with type 2 may also show high levels of insulin in their blood, while those with type 1 will show none. Type 2 diabetes, in particular, is harder to identify, and people can go about their lives for months without recognizing its presence. Those with a family history of diabetes are more likely to develop it later in their lifetime. Obesity, inactivity, high cholesterol, and high blood pressure also indicate higher risk. The treatment goal of type 1 and type 2 diabetes is to regulate and control normal blood sugar levels. Patients with type 1 diabetes are recommended to exercise and follow a diet consisting of the healthy, complex carbohydrates present in fruits, quinoa, brown rice, and vegetables. They should avoid foods high in simple sugars like candy, soda, white bread, and pastas, as well as trans fats. After every meal, type 1 diabetics must use insulin injections to regulate their blood sugar. Primary treatment of type 2 diabetes is exercise, weight loss, and a diet of complex carbohydrates. If that plan fails to regulate blood sugar levels, the alternative is prescription oral medication. Patients with type 2 diabetes only receive insulin injections as a last resort. Diabetes mellitus is a chronic disease where an individual's cells do not take up glucose. In type 1, white blood cells attack the insulin-producing cells of the pancreas, causing no glucose to enter the bloodstream. While in type 2, the insulin protein does not bond properly to its receptors and in a condition called insulin resistance. Some people are genetically predisposed to diabetes, but may only be expressed after the individual is exposed to an environmental trigger. This chronic ailment can lead to health concerns such as heart attacks, strokes, blindness, and kidney failure. But with proper treatment and management, those with the disease can lead to full, happy lives.